Welcome. So I will continue the tradition of introducing oneself. So I'm Greene Scientist on Twitter. If you want to tweet at me, that's probably the most important thing. I'm also a professor at the University of Colorado's Anschutz Medical Campus and the chair. That's OK. I have a small child who's only partway through vaccination, so I'll keep it on. Can you hear me OK? OK. And chair of our new department of biomedical informatics. I will aim to be efficient with this talk, so I may skip one or two things. So I'm going to talk about sort of integrating omic data across kind of data sets and layers. I'll just sort of think about why we might want to do that. So why do we ever want to integrate data sets? Why might you want to integrate data types? Do neural networks solve all of our problems? We hope the answer is yes. And then how does this actually relate to health? And so I just want to kind of start with a thought experiment. That's something that we experience sometimes. Imagine that you could do any genome-wide experiment and see the same result. That would be kind of weird, right? It doesn't matter what you perturb. And you might think, well, how could that possibly happen, right? There's no way you could do that and have that possibly happen, right? It actually matters what experiment you do. If you want to see what you see depends on what perturbation you made to your system. So you'd say, OK, there's no way that actually happens. But I have disappointing news for you, which is that this does happen. So there are some pathways. This is looking at RNA-seq. Actually, I think this paper is looking at microarray experiments. But essentially the same thing. You look at how genes are expressed across the genome. And there are some pathways that are repeatedly observed as differentially expressed, regardless of what the experiment was. And you might say, well, that's just because people aren't doing their statistical analyses correctly. They didn't control for background correctly. Well, this is actually if you do an appropriate control, knowing that some pathways have more genes and they sort of have different characteristics in them. So yes, there are some pathways that, regardless of the perturbation applied, show up as significantly differentially expressed in experiments. So that's kind of wild. And it's not just pathways. So you might say, well, pathway analysis is pretty tricky, right? Pathways are all different. They have sort of different gene membership. But this is actually true at the level of individual genes, too. So this is a paper from Jesse Gillis's lab at Cold Spring Harbor, where Megan Crow essentially curated 1,000 different experiments and performed differential expression analysis across them, and found that some genes could be predicted as differentially expressed, even if you knew nothing at all about the experimental design, which is kind of fun. So sometimes you say, like, wow, I did this experiment, and someone else did that experiment. We saw the same genes as differentially expressed. That must mean there's some hidden connection between these systems. Well, that's not necessarily true, because there are just some genes that show up all the time. And you might say, well, then we should just filter those, right? We should just say, well, whatever those genes are that are always differentially expressed, we should just get rid of them. So if you were going to do it through the traditional approaches, what you'd actually have to do is curate, every time you want to analyze your data set, you just have to curate 1,000 data sets that look just like it, perform the same analysis on your data set and 1,000 other data sets, and then you could just subtract out the things you saw in all the other data sets. You can imagine this could be quite hard. And you can come up with ways to make it slightly more efficient, like maybe there's a library of curated data sets that people can use, and you can select which ones are appropriate. But at the end of the day, there's no way to sort of tackle this problem that's not just an enormous amount of work if you want to do it with curation. So this is something that Alex Lee, who was a PhD student in the group who just graduated, was sort of looking at and recognizing that one of the technologies that she developed for a different problem earlier in her PhD could potentially address this one too. So what she had been doing was training variational autoencoders on gene expression data to be able to generate new synthetic data. And so the way this works, she has a whole bunch of data. She trains a neural network to put it in some low-dimensional space. And then she trains another neural network to take data from that low-dimensional space and make it look like the original data. So essentially, you have this sort of compression that happens and then a reconstruction. And one of the keys of this specific technique is that the low-dimensional space follows some predictable distribution. In this case, a Gaussian. And so then all she has to do, if she wants to create a whole bunch of new data, is just sample from that distribution. And so she can create arbitrary new gene expression data sets that did not actually exist, but that look like data that could have actually existed. And so you can do this. So this is one of these is real. And one of these is an experiment that she generated. The way that her technique works, there's a template experiment. So she does this sort of in the low dimensional space of the autoencoder. She does this translation in that space and then can generate new data that have essentially the same distance relationships in reduced autoencoder space. So one of these is a real experiment. One of them is a synthetic experiment. It doesn't really matter which is which, but it's just kind of key that they look vaguely similar. And they kind of have sort of vaguely similar effect size, even though sort of the perturbed genes are largely different, except for the generic differentially expressed genes, which continue to be generically differentially expressed. And it's also, if you collaborate with me, you should know I'm a horrible person. So the first thing we did, this was an experiment in a microbe Pseudomonas aeruginosa. Our collaborator, Deb Hogan, has been collaborating with us for probably 8 or 9 years on this type of problem. And so I just sent a message to her lab Slack that said, hey, can someone let me know what this experiment was perturbing? And they came up with this really wonderful story, which is a great, nice bit of sort of biology forensics as to what the experiment might have been. But one of the key things to know is that this experiment doesn't actually exist. So it's a completely synthetic experiment. And so once you have this technology, then you can do something like say, hey, does this thing actually work? Like if I simulate generic experiments that never actually existed but that might have, are the differentially expressed genes in those generic experiments the same as the generically expressed genes in actual curated experiments? And so what I'm showing you here on this axis is the differential expression prior score from the Crow et al. Paper. So this is from curating 1,000 different microarray data sets by Megan Crow and Jesse Gillis's lab. So that's the paper I showed you before. Every gene has a score. Those go from highly generically differentially expressed to not often observed as differentially expressed. Then there's a method that Alex invented that she called SOPHI, which stands for something like all methods in bioinformatics, but in this case, let's just know it's called SOPHI. So this is trained on RNA-seq data. So this is from Recount 2. So this is human RNA-seq data. This is human microarray data. The brighter or more intense these spots are, the sort of more genes fall into that. And what you can see is there's a really nice correlation relationship where things that are observed as not often differentially expressed in the Crow et al. Data are also the same in sort of Recount 2 with the simulation. And the things that are more often observed as differentially expressed in Crow et al. Are also often observed in the sort of synthetic data. So what this means is now you can simulate gene expression data that didn't actually exist. You don't have to do the curation. This works for arbitrary experimental designs, which is kind of nice. And you can plug it in with sort of any analysis strategy. So if instead of imagining sort of a two-condition differential expression experiment, you might imagine there are some genes that are only observed as differentially expressed if you have some like dose-response experiment. You can sort of do that same type of analysis here. So the first question I asked was, why might you care about integrating data sets? And when I was here in 2019, or actually maybe even 2018, my answer was because we really care about the things that are the same across many different data sets. And of course, now it's been 3 years. So when I returned, I'm telling you, well, sometimes you care about the things that are the same across many data sets, but if you start looking over and over at the things that are the same, it gets a little bit disappointing because you see the same thing, regardless of what conditions you're looking at. So sometimes it is important to look for consistencies, but sometimes it's also kind of nice to look for inconsistencies, right? What are the things that are unique to a specific data set or unique to a specific context? And I actually think that's where the sweet spot is. You really want to see things that are sort of not data set specific, but are context specific. And so this is kind of one path to sort of integration and how it can help you learn. This is available on GitHub if you want to play around with the code, if you want to synthesize your own data and use it to analyze your own RNA-seq experiments. And there's also a manuscript kind of working its way through peer review, but that's also on bioRxiv if you're interested in reading about the technique. OK, so sometimes you want to integrate a whole bunch of different experiments of the same type so that you can learn something new about sort of that data type. But often I get, we work mostly on the transcriptome. And what I often get asked is, well, why do you work on the transcriptome? Why not pick some other ohm? There's got to be the right ohm, right? What's the ohm we should all work on and just ignore all others? So this is a project that Jake Crawford, who is a PhD student in the lab, was working on. And really the starting point was just this question, if you've got to pick an ohm, which one is best? And so in this case, we're looking at sort of trying to understand the effect of somatic variants in cancer omics data sets. We use the TCGA data sets from the Pan-Cancer Atlas freeze in this project. And what we're really trying to do here is just ask which one has the most information to predict somatic mutations in key genes. This was previously published by a PhD student in our group who's now moved on and done a bunch of other interesting stuff. I'll give you a brief idea of kind of what he's doing there. The input data is just a whole bunch of different omic layers. And then the endpoint is trying to predict the presence or absence of somatic mutations in 268 cancer-associated genes. So the way we framed this problem was really simple. We kind of imagined that you had some key regulator that you knew the status of, and you wanted to sort of take cases where you knew there was a variant and cases where you didn't know if there was a variant and use those to build an expression-based classifier of whether or not the regulator was present or absent. One of the cases that we used as sort of the initial analysis for this was P53, which has really nice transcriptional signatures, so it's easy to detect. And so what we essentially do is train a classifier with samples with P53 loss labeled as positive examples, samples without any known P53 loss event as negatives. If you do this, you can get a nice gene expression classifier. There's a whole bunch of work in a series of papers sort of demonstrating. So in this, you can in a couple of these papers, we really do a nice job of demonstrating that you can find phenocopying events. So things that don't have the canonical mutation, but that have a different mutation that seems to be inducing the same cellular state. We identify a silent variant in P53 that actually appears to alter splicing in a dominant negative fashion. So there's a bunch of sort of stuff validating this strategy. It's not terribly important here, except to know that sort of this is a strategy that you can use if you want to kind of predict the activity of key cancer genes, and it's really pretty straightforward to implement. And so what Jake then did is took that strategy and just applied it at scale to 268 different kind of cancer-associated genes. And in this case, what he pulled first was sort of the genes with the most mutated in TCGA. And what we're showing here is this is the log 10 adjusted p-value. And then this is the AUPR for a data set where we've left signal in with compared with one where we've permuted the data set to remove signal. And so essentially, this is trying to control for mutation frequency abundance, that sort of thing. And also the different distribution, the permutation retains cancer type information. So it controls for the gene expression, the different distribution of mutations across cancer types. So that is an important element. So that's kind of the baseline that you're comparing to. And you know, it's always nice to include some controls when you do an analysis. And so what this is comparing here, this is if you just pick 268 random genes. This is if you pick the most mutated genes. This is if you pick this curated set of genes. You know, how many of those are predictable based on, in this case, gene expression data alone. And you can see that sort of as you enrich for sort of cancer things where you would expect there to be a cancer-related signal, you see that, in fact, more of those genes are actually predictable. So these somatic mutations in the cancer-related genes are more predictable than in random genes. Let's see. I think, yeah, OK, here we go. And so the next thing we wanted to ask is, OK, what data types give you the sort of best prediction quality? And so this is showing gene expression data. So this is the RNA-seq expression data out of TCGA's Pan-Cancer Atlas. This is 27K methylation data, 450K methylation data. You can see they're kind of generally really similar. There's not that much of a difference here. There's a lot of kind of, generally the scale of the signal seems to be about the same in each of these. And then another thing that's kind of so then you can actually set them up to contrast. So you can compare methylation versus expression, for instance. And we do see that generally gene expression, so RNA-seq data, was the most predictive layer. But it's not that strong of a predictor, right? I mean, the difference between these two is not that high. If you look at how many total genes are predictable and you compare it to the ones where there's a difference, it's really not that different. And then finally, what we wanted to ask was, OK, but is this really driven by sort of which genes are predictable, and it doesn't matter what omic layer you picked? Or is it driven by some omic layers are better at predicting the sort of state of some genes, and other omic layers are better at predicting the state of other genes. And so this was our attempt to get at that question. So what this is showing is a heat map. The brighter the square, or the rectangle in the heat map, the more predictive that data type was. And what we were kind of imagining here was two options. One option where you'd see kind of bright elements kind of scattered throughout. And in that case, not sort of in lines, but just kind of randomly scattered throughout. And what that would tell you was that you had to get exactly the right ohm, and you had to get exactly the right gene to find that functional signal. The other option is that you'd see stripes, essentially. If you see stripes, what that tells you is, it almost doesn't matter what ohms you pick. They're all kind of getting you the same signal. It's just kind of different regulatory layers. And we see a lot more things that look like stripes than kind of these like punctate marks. And so what that's kind of telling us is, it sort of doesn't exactly matter what omic layer you end up profiling. You're going to capture a lot of the same functional signal, where functional signal is described as kind of the downstream consequences of somatic mutation events. And so this is kind of liberating if you're thinking about your study design in cancer omics. If you're really interested in trying to capture this kind of functional state induced by somatic variants, you can probably pick whatever. If you have to pick, you're probably better off with RNA-seq data, but sometimes it's pretty hard to get RNA-seq data of high quality given sort of sample collection characteristics. And so in those settings, if you need to profile methylation or something else, really a lot of the same signal is there. And I didn't show you the RPPA, microRNA, or mutational signatures results as we were kind of zooming through. But if you want to look at the paper, that is actually, all those results are actually in the paper. And then the second question that Jake wanted to ask was, okay, so individually it looks like there's a lot of redundancy. Can we actually learn anything if we integrate across omic layers? And so he implemented a couple relatively simple methods to just ask, okay, is there like a free win? Like putting data together from different omic types gives you better performance. And so he put this together in a few different ways, using logistic regression, multi-layer perceptrons, the like. And what we ended up finding was there really didn't seem to be much of a benefit from integrating data from multiple omic types. So it at least wasn't a free win. It might help, but if it's going to help, you're going to need much more sophisticated methods for it to be successful. So this was just showing for P53. Over a whole set of genes, we could ask if kind of the best single omics predictor was better or worse than the best multi-omics predictor over multiple runs. And what we ended up finding was there really didn't seem to be much of a difference there. So we kind of inherently believe that these multi-omic predictors should be better. But what we found was not strong evidence that multi-omic predictors are inherently better. So as we're sort of seeing multi-omic efforts, it's really good to kind of understand what baselines those have been tested against. And in this case, we really didn't see a benefit here that was convincing from the multi-omic integration. And so this paper just came out like a week ago. If you're interested in genome biology, of course, the GitHub has been up for a couple of years as we've been doing this project, as is true of most of our stuff. And so if you're interested, you can take these, the GitHub repository that's linked here and sort of plug in your own methods. You can plug in your own assumptions and kind of see what you end up seeing in your own analyses. But I was kind of, I was really interested in understanding the results of this kind of relatively large-scale benchmarking project. And it was kind of intriguing to see, that some of our initial assumptions, like IDH1 in low grade, IDH1 being more predictable from methylation than from RNA-seq data, didn't really get borne out in the data. It really looked like the redundancy levels were much higher than we expected. So if you're interested, this is also available. So there was one project that I dropped in as a link for a paper that I'm not going to talk about because it was going to take too much time to go through, so I pruned it beforehand. But if you're interested in other types of data integration where we see a little bit more benefit, since I've just told you that we see a lot of redundancy and not a lot of benefit, this is a paper from, that was led by Milton Pividori, who's a postdoc in the group, where he essentially learns low-dimensional representations from large collections of public data and then projects genetic associations, sort of represented as TWASs, through these low-dimensional representations, and does see some benefit there. So while I'm telling you that it's not always a free win to integrate data modalities, I will say that in some cases it does appear that there are benefits. And there's a lot of benefits from an interpretation point of view of this type of strategy. So we've kind of talked about why we might integrate data. Sometimes we do see benefits, sometimes we don't see benefits. It really does matter how you actually do it. But we always know that kind of sitting on the horizon is deep learning, which is this tool that's going to save us. We're not actually going to have to think anymore. We're just going to plug in our results into multi-layer neural networks, and we're all done. So this is a project that Ben Hale, who's a PhD student in the group, has been working on for a little while. This actually started as a new methods project, and then essentially, as all new methods projects do, worked its way back to trying to learn something about why the methods didn't work. And so that's essentially what this is. So what he ended up doing was taking multiple different data sets, in this case recount3, GTEx, and then a simulated data set, applying multiple different models, logistic regression, 3-layer neural network, 5-layer neural networks, and predicting for different problems to try to understand what were the features, what were the characteristics of methods that led to good predictions. And so this is the really fun part of the talk where I get to tell you nothing he did mattered. Essentially, the methods were irrelevant. The problem mattered. So your prediction performance does depend on data set, and it depends on the problem. But the methods were essentially wholly indistinguishable from each other. He did a whole bunch of things where we thought they would help. So this is actually the x-axis here is as you increase the number of training samples. It was really surprising to us that the curves looked so consistent. We did things like pre-training neural networks. That didn't really seem to make much of it. It makes a little bit of a difference, but it doesn't sort of change the relative ordering of methods. Essentially, changing the relative ordering of methods was pretty hard, and I would view the relative ordering here as pretty indistinguishable from each other. If you have to pick, logistic regression looked fine. It's kind of my takeaway. So you could take this set of results and the 10 more in the paper like it and just say, well, OK, all signal is linear. We don't have to worry about nonlinearities anymore. They don't matter. They don't exist. And so that would have been a good point to just quit and write the project off and go find something else to work on. Ben did this experiment, which I thought was pretty clever and very exciting. So then he just took his endpoint and then essentially corrected data for the endpoint using a linear correction. So he essentially removed linear signal in the data so that we could see if there was any residual nonlinear signal that persisted even after you removed the linear signal in the data. And so what you can see here is that if you look at logistic regression, which is the dotted green line, if you remove the linear signal in the data, logistic regression no longer works. Probably not terribly surprising since that's what it's trying to predict from. But these nonlinear methods still work, right? There's still some residual signal in the data. So there is residual nonlinear signal that exists in the data. For some reason, it's either the nonlinear signal is redundant with the linear signal. So if you have the linear signal, you don't need it to make predictions. Or it's just so subtle that it's hard to take the methods that we're using right now and sort of use it to get a predictive performance that goes above and beyond what we see with the linear methods. So our takeaway at this point is there is some nonlinear signal. It doesn't seem to help prediction performance, but it is there. And so then the question kind of arises, is it redundant, or is it just that our methods aren't quite appropriate for the problem? That one, it's hard to argue the methods aren't appropriate for the problem when those same methods can make predictions on the sort of the nonlinear residuals, right? So we think it's probably redundant. But a lot of kind of open questions. This also, like everything I've talked about, is available on GitHub. If you're interested in playing around with it, I think this is a really fun project to kind of pull off the shelf and okay, but I have you just tested a couple methods, but I have my favorite method. Does my favorite method fall into the same category? And so this gives you a really nice benchmarking set to use. There's also kind of some challenges that come up when you do this type of work, like how you actually hold things out. Do you hold out a data set at a time? Do you do sample-wise holdout? So there's multiple different holdout strategies implemented here. We do think you should hold out the data set at a time, and we do see kind of performance inflation in other settings. But none of those really affected the underlying results. And so, yeah, I think this is a really nice kind of, if you're thinking about trying to make a predictor from gene expression data to do something, I think using this repository and kind of the benchmarks that are in it is a nice place to get started to just sort of understand the performance of your method in comparison with other methods. OK. So I really like every once in a while to read some relatively old stuff on biomedical research or kind of scientific research in general. And Vannevar Bush wrote a letter to FDR that essentially laid out the case for a national infrastructure for science after World War II. And so the academic system that we're all in right now is essentially built and envisioned in this letter and sort of by this strategy that Vannevar Bush developed. So if you want to know why we do biomedical research, why it's funded the way it's funded, sort of that's essentially his vision. And he ended up writing in that letter, the pioneer spirit is still vigorous within this nation, and then I've adapted it. Open data offers a largely unexplored hinterland for the pioneer who has the tools for their task. I really do think this is the case. If you are willing to pull a whole bunch of data off the shelf and put it together, if you think pretty carefully about how you do it, you can learn a lot about that. And I think you can learn more than you can learn from individual data sets, even very large-scale individual data sets. And so I really think the practice of science is kind of about linking together these disparate data to make sense of the world. So I really like, I like this framing. And if you really do want to understand why biomedical research is structured the way it is, with grants being given to universities as opposed to like being done in government labs, I think sort of seeing Vannevar Bush's vision is reading this letter is kind of interesting from that historical perspective as well. So at the end, I just want to sort of think about, okay, but how is this actually going to impact medicine, right? We've just talked about a whole bunch of esoteric research problems, but none of them seem to necessarily have a real tangible impact today. I think as a field, kind of fundamentally, I like to say our business is serendipity, right? Like what we're trying to do is take the thing that's indicated in large collections of data and reveal it to someone so that they can take an action they wouldn't have otherwise taken. Pasteur essentially wrote in the fields of observation, fortune favors the prepared mind. And I think basically what we need to do is develop ways that sort of take these unexpected patterns in data and make them happen sooner, like really make that routine. And there's a lot of ways to do that. But I think for our field, this is fundamentally where the power lies, right? Is being able to help people make better care decisions and sort of intervene at the point of care. This is a story from our hospital system, UC Health, where one of the programs that our department helps with and a lot of our faculty play a part in is our Center for Personalized Medicine. So we have a preemptive PGX program. And so like literally the clinical decision support is built into the EHR so that if someone is prescribing a medication and there's a variant that they have that would alter the metabolism of that medication, there's like an alert that fires, it suggests a different course of treatment or a different dosing. And I think the stuff that I talked about today is definitely not here yet, but if we do it right, these types of omic profiles, these types of kind of pattern recognition problems are also going to work their way ideally into the electronic health record. And that's really kind of where we can transform the care that people are receiving. I want to take a very brief moment to advertise CU Anschutz as a place to do this type of work. I know I'm at UCLA, and I know you have wonderful stuff here too, but I just want to emphasize, we have a really great research environment. We just started a new department 10 days ago in biomedical informatics, and we've got to hire about 20 faculty. And so I'll just put this up here. If you haven't thought about the University of Colorado, please do. Before I get pulled off the stage, I'll now move to the next slide.
You mentioned who's the chair of that department?
It embarrasses me to mention who's the chair of that department, but I am the chair of the department, so if you... So I wasn't going to mention that, but if you have questions, please come talk to me. With that, I want to thank the folks who make this possible. So the research that I talked about today was led by Alex Lee, who just graduated, Ben Hale, Jake Crawford, and Milton Pividori. From the funding point of view, we really appreciate the folks who give us money, and if I have done this right, there will be a little bit of time for me to answer questions. I have done this right? Maybe?